Greetings, everyone. I chose this title to be a little bit provocative. Neuroscientists are a little bit jealous of physics because physics has so many laws, and neuroscience doesn't. But we have one very important one, which is the Weber-Fechner law. Most of you, or probably everybody in this audience, knows about the Weber-Fechner law. This is a log rule that describes our subjective perception, and it describes it as a proportional to the logarithm of the stimulus intensity. This is a overriding general rule that applies to perception, but it's also present in many, many other things in the brain. For example, it applies also to our space and time perceptions. And I'd like to convince you today that the reason why this perception occurs is because there is a dynamic in the brain that follows these log rules. And I would like to convince you that synaptic weights, firing rates, population synchrony, and many other dynamical features in the brain follow this rule. And the reason why they do that, or perhaps the foundation of this, is that this dynamic rides on a structure where the macroscopic, the microscopic, and mesoscopic network levels also follow the log-normal distribution rule. Well, if you look at the most famous or perhaps most dominant dynamics that you can record from the human brain is the EEG. The EEG comes into various classes, and that also follows the log rule. There are these many classes that you can see here that were classified by many different groups in neuroscience. They fall on the integers of a natural logarithm scale. And they are held together by a rule which is called cross-frequency coupling. So this is the first dynamic rule that has a log relationship. And I'd like to go back now to the foundations of the structural foundations. Not so recently, not so long ago, a very prominent study was published by the Allen Institute, and it showed that connectivity in the brain also follows log-normal distribution. What it means is that any part of the brain, any part of the cortex is connected to a handful of other structures very, very strongly. But the same structure is connected to many, 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 many other structures, many, many other regions weakly. And distribution shows a log-normal rule, no matter whether you are looking at cortical-cortical, cortical-subcortical, or the whole brain, ipsilateral or contralateral connections. Now we go from the macroscopic connections to the microscopic connection, all the way down to the axons first, and we will see that the diameters of the axons are also broadly distributed. Most of the axons have very small diameters, but some of them are 100 times bigger than the others. And this rule is present in every mammal that has been investigated so far. Now we go even further down to the microscopic features, to the level of the spines. And when we measure the spine distribution of a CA1 hippocampal pyramidal cell or a layer 5 cortical neuron, then we find that that shows a beautiful log-normal distribution again. So spines, of course, are the recipients of synaptic activity. So we would like to know whether the synaptic size, as we know, is correlated with synaptic strength, but we can measure synaptic strength also relatively directly. You can do it in a slice preparation, or in vivo, what you can measure is the spike transmission probability, which is a proxy for synaptic strength. So I'm showing you here 3 different scenarios, the synapses, and then through these different synapses, the probability of transmission is, the first one is very high, and the third one is very low. And when we plot them, we will find that independent of the brain state, synaptic transmission follows this log rule. And moreover, those synapses that are strong in one state, they will be strong also in the other states, and the relationship will be proportional, even though the overall strength can change between, let's say, slow wave sleep or when the animal is running around in the environment. So that's the first and interesting, important rule, that is the macroscopic connectivity, microscopic connectivity, and the macroscopic physiological changes such as spike transmission probabilities are also affected by this rule. I tell you another level, which is the firing rate distribution of neurons. In summary, if you take 1,000 neurons from any structure in the brain or the spinal cord, and you look at their firing rate distribution, it's like the fingerprint. Every neuron has a fingerprint based on its own firing rate. Most of the neurons like to fire at a low rate, a minority fire at a high rate. And I'm talking about not different types of neurons, I'm talking about neurons within the same exact class, such as layer 5 pyramidal cells or CA1 hippocampal pyramidal neurons. And we did this game in the entorhinal hippocampal system. You can see here multiple regions, and the distribution of the firing rates in the multiple regions show the same rule. The shape of the curves are slightly different, and the firing rate distribution during non-REM sleep and REM sleep, these are the extremes. They are also different. Nevertheless, the shape of the distribution is the same, and Lognormal rule applies to all of them. Now, it becomes more exciting when we are asking, what is the firing rate, the long-term firing rate of a neuron in one environment and in another environment? Those of you who are familiar with the hippocampus know that hippocampal cells are sometimes called place cells because particular pyramidal cells like to fire in a particular part of the environment, such as this is a rectangular box, and this neuron had a peak firing rate somewhere in the middle, and had another one here. The same neuron now fires at a particular segment of a linear maze where the animal is going to the right, but when the animal returns and runs through the same exact spot of the environment, it doesn't. These are the place cells. Now we can look at the relationship between the place cells in one environment or in a linear tract where the animal is running one direction in the other direction. And it doesn't matter which direction to look at. And even though these colors represent that, oh, this neuron fires a lot in one direction and it doesn't in the other, and it fires more in one environment than the other, at the log scale, remember in log scale, 10 is only one unit, then there is a nice relationship. This is only one single session that we recorded from in the rodent. But if you look at many neurons, you see that the relationship is very robust. In other words, those diligent neurons, they fire fast in your brain now. The same neurons will fire fast tonight and tomorrow and for a long time. I can't say forever, but probably for a very long time. And it applies to other regions in the hippocampus, such as the CA3 region. Now, it's not only firing rate, but another firing pattern is called the burst, follows the same rule. And in this case, I show you that you look at the burst probability. That is, what is the probability that the neuron can fire burst in half an hour or 10 minutes in one environment, such as box A, and then you put the animal back in to put in another environment, and it shows that the propensity of this neuron will remain the same. Some neurons like to burst a lot, doesn't matter which environment they are in. Others, the majority of them, like to fire burst only rarely. And of course, this applies not only to to this situation, and the firing rate distributions are becoming exciting when you ask the following question. Can I predict a arbitrarily chosen hippocampal neurons firing rate in a novel environment or a old environment if I record from this neuron for a long time during sleep? And the answer is yes. So this is what is shown, that this is the x-axis shows the firing rate distribution of many neurons in the home cage. And then if you look at the firing rate of the same neurons in a maze that was completely novel, the animal has never been there. And we can do this before and after the exploration. This is pre-exploration sleep, this is post-exploration sleep, and you will see that the prediction is pretty reliable. And of course, this rule applies not only to the hippocampus, but to many other brain regions. In fact, there is no exceptions to date. There are some examples from the mouse barrel principle cortex, principal cells of the mouse barrels, and monkey neurons in various regions. And this is the same even if you are looking at the firing rate distribution of the ventral horn neurons in a different species, such as a turtle. So this was about single neurons. Now let's see how neurons behave as a population. This is a hippocampal pattern with many neurons recorded simultaneously, in this case 75 neurons recorded simultaneously. And every now and then when the animal falls asleep, there are these patterns, they are called hippocampal sharp wave ripples. And this is the most synchronous pattern of the mammalian brain. So you may wonder, what are the rules here? What is the probability that neurons fire together in this time windows of the sharp wave ripple? And the answer is that it varies a lot. But variability is such that most of the time only a fraction of neurons fire, but sometimes many more. And that shows a beautiful global distribution also. So far, this was only about structure and dynamic separately. Now let's try to bind the two together. So this is a nice study done by the Klausberger group in Vienna, Austria. And the finding is the following. Ventral hippocampal neurons project to some areas, such as the medial prefrontal cortex, the nucleus accumbens, and the amygdala, and different sets of neurons send their axons to these different regions. But some of them send axons to all three of these regions. That is, they have longer axons. And it turns out that neurons that have longer axons fire 3 times more, in this case, than neurons that have shorter axons. So structure and function are related very strongly to each other. So far, I showed you a laundry list of things, and you may wonder whether this is exciting or not at all. Maybe it is just an interesting distribution that is present, of course, in most living systems and perhaps even non-living systems. Is it a statistical curiosity only, or it has some more meaningful, deeper significance? You may say that it is only a statistical curiosity. My take on this is that's probably not the explanation, but perhaps it's the brain's attempt to reconcile conflicting demands among wide dynamic range, as we know, is there, stability, robustness, plasticity. Plasticity and stability are fighting with each other, and the solution in nature when it's trying to solve complex problem is always diversity. And diversity is what is beautifully represented by the log-normal distribution. So now let's see a couple of examples of this log rule, or skewed rule. Probably that's a more general form, because sometimes it's not log, sometimes it can be described by a gamma distribution. This is a term statisticians understand. And for example, if you look at or you ask the simple questions that how many tuning fields a neuron can have. In the hippocampus, a question has to be posed like this: how many place field a neuron has? Most of the neurons have only one. Some neurons have 2, 3, 4, and rarely some neurons have 5. And you can see that this distribution shows a very nice rule-like pattern. Same thing you can ask is that what happens if you put the animal in a very large environment and the animal runs through this very long 50-meter long track, then most of the neurons will have a place field only at one spot. But some neurons, such as this, can have 25, 30, and then the distribution is skewed, as is shown here. Or if you train the animal to run on a radial arm maze, and you ask, if a neuron fires on one arm, what is the probability that it will fire on another arm? Then it is a law-like distribution. Even more exciting is perhaps that you can ask, if a neuron fires in one environment, what is the probability that it will fire in another environment? In the hippocampal literature, this is called remapping, showing that, and the claim typically is that when you put an animal into different environment, different sets of neurons fire, and they are orthogonal to each other. This orthogonality or independence is true for the majority of the neurons, but not to all. As you can see, this neuron, for example, or this group of neurons here, they fired in all of the 11 rooms that the experimenters tested. Some neurons only fired in 4 environments, and you can see that the rule is pretty much the same. So this is an interesting thing, of course, because if the hippocampus is recognizing different environments as different, then of course we need something that recognizes the similarities also across environments. When you enter a new room, you don't say that everything is new here. In fact, it takes for a while probably to say whether this is distinct from the other and explain why. If you go to a Marriott hotel room or a standard chain hotel room, then the room layout and the features are pretty much identical whether you are in Shanghai or whether you are in New York. So there are features and there are elements in the neural networks that are capable of generalizing and distinguishing. And the 2 ends of the distributions are probably doing that. The high firing rate neurons, which is this end, are the generalizers, the specific ones, the specifiers, the ones which low firing rate neurons. So as it turns out, the reason why this could be the situation is because there is a relationship between the firing rate of the neurons, which is the propensity or the feature of the neuron, and what it actually represents. In other words, those neurons that have multiple place fields or they have larger place fields, they fire faster. And remember, I just mentioned you that neurons that have longer axons also fire faster. Those neurons that are these diligent ones, they like to talk to each other. They exchange information among themselves and they receive information from the majority of the low firing neurons. In other words, they form what we can call a Richman club. These are the club neurons that form nodes and they control the system as well as get information from very large part of the system. So if I could convince you that there are physiological meanings for the distribution of strong and weak, this is shown here, this synapse is many, many times stronger than the other one. Whenever it fires, whenever it sends an information, its vote, it says something more important to the postsynaptic neuron than the synchronous firing of many weakly active presynaptic cells. So if this is an important thing, then that has to be regulated somehow. So in the second part of my talk, I'd like to discuss with you how these long distributions are maintained. And of course, if the important thing is that to demonstrate that these are indeed useful for function. So the first thing that we are looking for is what are the mechanisms that maintain the 2 ends of the 2 extremes of the log firing distributions. What you can see on the left lower part here is the firing patterns, the firing rates of the neurons, and their sequential firing during these so-called up and down states of slow wave sleep. During slow wave sleep, many neurons fire together. This is called the up state. And in between, most of them, or nearly all of them, are silent. This is called the down state. And during the down up transition, some neurons start firing. And it turns out the neurons that are the leaders are the fast firing neurons. And this is true for the first half, second half, or if you have quintiles, then it's true for all the quintiles. In other words, it is a tendency that the fast firing neurons tend to initiate the sequences all the time. Now, this is an interesting thing because now you can ask, what is the relationship or co-firing relationship or synchrony between neurons in a system with such a wide firing rate distribution? The answer is that neurons that fire fast will coincide with each other in time, with a high probability. This is, of course, not a surprising thing. Even Poisson neurons do that. Neurons that are silent or fire, rarely their coincidence or cross-correlation will be low. This is again trivial. What is not trivial and is not present when two neurons with Poisson spiking distributions are compared is that the probability that the fast and the low is firing much, much less than the low firing neurons. And the reason for that is exactly this distribution. There is this rule that at the down-up transition, which is the most prominent firing pattern during sleep, is that this fast-firing neuron fires early, the slow-firing neuron fires late, and they are segregated in time. That's why they don't fire together. Now, if you are a believer in spike time independent plasticity, this kind of rules, then the consequence of this is that the fast neurons that fire fast and have this coincidence, this distribution, this anti-correlation with each other, with many repetitions, they get weaker and weaker, and the fast-firing neurons become slower, and the other end is also true, the slow-firing neurons become faster. So what happens during sleep is that there is a renormalization of the distribution of the firing rates. During waking, while we are attending this meeting or discussing with your friends in the interim, the firing rates are getting wider and wider. Slow firing neurons become less active. The fast firing neurons become more and more active. But this will be normalized tonight when you will have your several hours of non-REM sleep. So this is a rule that ties together or maintains the distribution of the log-normal pattern. Now, are these distributions useful, and can we affect them with something such as learning? So we approach this question by using the so-called almost a standard task in the hippocampal field. We train the animals in a novel task, but before we do the training, we record from the same neurons before, during sleep, and after sleep, and ask, in our case now, what is whether there is a change in their cross-correlation functions. So this is a matrix of neurons that start here from the low firing to the high firing pyramidal cells. And this is the same with the other neurons. So every single cross-section here is a cross-correlation function and result for all the neurons. And you can do the same game after the animal explored a novel environment. And now we can compare the two by simple subtractions. And what you will find is that the changes as a result of learning didn't occur anywhere randomly. The plasticity occurred in the low firing neurons. A neuron, which is pretty silent most of the time, but when it spikes, it says something important, and its probability can be changed by learning. We can call these neurons plastic neurons, so they are slow firing neurons, and the fast firing ones can be referred to as as rigid neurons, because they don't accommodate and don't change as a result of learning. So just summarize here a little bit, and we can say that the distribution of the firing rates, for example, are very strongly cued, which means that half of the spikes are contributed all the time by the same small fraction, let's say 10% of the neurons. But the other 90% of the neurons are also very important, because they contribute the other half of the spikes that are present in any time window in your brain at the moment. And I suggested that the right hand of the distribution, the fast firing neurons, are the generalizers, and they are active in most situations, whereas the left hand distribution is probably useful in learning situations. And you can say that, you know, a small fraction of neurons, that is the generalizer neurons, are consisting or comprising a good enough brain, because they are ready for you all the time, giving you a good enough answer and help out in most situations. However, if you would like to solve something or understand or perceive something with 100% accuracy, 100% of the time, then the brain has to mobilize a very large resource of the neurons, including the slow-firing neurons. And of course, it also implies something very meaningful, namely that when you are talking about novel and familiar, these are words made up by humans. These are distinct, discrete words, but nevertheless, the distribution in the brain is not discrete, but continuous. So one assumes the other. Every single novel situation contains elements of familiarity. There is no new river, there is no new mountain. All mountains that you will ever see has elements of previous knowledge. That suggests that indeed there is some pre-existing structure in our brains that are due to these generalizing minority neurons. I jump forward and I say, well, if these distributions are so important for the cellular network circuit organizations, and as we started, this is so important for our perception because this is the Weber-Fechner law, is there any intermediate mesoscopic pattern that should or would follow the same rule? So again, I'm showing you this hippocampal sharp wave ripple. These are the most synchronous patterns in the mammalian brain, and they are believed to carry information from the hippocampus to the neocortex. We have about 2,000 to 4,000 of them every single night. And when we look at the duration distribution or the distribution of the durations of these events, we can see that most of the events are relatively short, about 20 to 50 milliseconds, but some of them are longer than 100 milliseconds. Does it make a difference whether you have a long or short ripple? Yes, it does, because when we look at the probability of these wider sharp wave ripples, then we find that they are occurring at a much higher chance in a novel environment. Also, when we are looking at them in a memory task versus non-memory task, we find that when the animal is under a memory or learning situation or learning demand, then the probability of the wider sharp waves is much higher. So if this is the case, then we can ask, well, there is one more example here showing that during correct trials, the sharp waves or the preceding sharp waves are longer, whereas in incorrect trials, they are shorter. So if this is a distribution, can we manipulate the brain, or can you manipulate the hippocampus, so particularly the CA1 region, and make average length sharp waves longer? We can use a manipulations that we can refer to as a closed loop optogenetical intervention, namely when we can capture a ongoing ripple event with its underlying firing patterns also, and add to this a prolonged artificial ripple by shining light to the pyramidal cells. Then we may, with this manipulation, we prolonged most of the sharp wave ripples to longer versions. Is it beneficial to the animal? And the answer is yes. Compared to the control groups, the performance, the memory performance of the animals is significantly better when we prolong sharp waves. Conversely, which I'm not showing here, when we abort or shorten the sharp wave ripples, then learning performance is impaired. And in this study, we provided an explanation for that, namely that what happens is that when the animal is faced with a choice of going to the right or left in a maze, an already started trajectory, I'm going to the left or I'm going to the right, is prolonged. That is, during the artificial synthesis or the synthetic ripple, the trajectory is not following a random pattern, but it continues the trajectory that is already started by the spontaneous version. So then we can come to this important part that is probably most relevant to this audience, is that how do these dynamic changes affect perceptions and experience? We can put the answers in two broad categories, and you can say, well, the way how it works is that when we are born, the brain is an empty structure and we can fill it up with information with learning. It is referred to, this framework is referred to as blank slate or tabula rasa. This is the dominant thinking in neuroscience and in AI. But some of us challenge the idea, and we say that the most important preoccupation of the brain is to maintain its own dynamic. The brain can generate myriads, many different preconfigured dynamic and many sequences and patterns. So there is no need to add more, because if you add more, you can destabilize the system. So you can generate a stable system with a vocabulary that from where you can choose and add significance to any of these patterns. And I will explain this idea a little bit in the next couple of minutes. So are there supports for each of them, or there are supports only for the tabula rasa idea? And as I said, this is the current thinking, that the brain is mostly random connections, which we know this is not the case. There is a Hebbian plasticity. There is an excitatory inhibitory balance, noise, and separate writing and reading mechanisms. And this leads us to this blank slate idea that is not a new thing, is not a unusual thing. In fact, this is probably the most dominant thing. And this is, of course, not only in neuroscience, but especially in the artificial intelligence community is very prominent. You build a AI network, and which is empty, and you fill it up with information, and this is how it becomes rich. And this is also the source of catastrophic interference. That is, the system collapses after too much information is provided. Nevertheless, it is adopted as a framework by many, and perhaps the reason for that is the giant of this field is Alan Turing. Endorsed it very early on, and it explicitly says that presumably the giant brain is something like a notebook. It's like a blank slate. But in fact, there are many other ideas, many thoughts, especially by my ex-students, such as Professor Harris, who showed that in the auditory system, during slow wave sleep, the number of patterns is enormous. There are many patterns to choose from. And in fact, when you apply different tones and natural patterns and any pattern, then they will not generate a new one. They will choose from the existing possibilities. And this is the same kind of thing that happens in the hippocampus. We can record a sequence of activity, let's say during running, the same sequence that I showed you a minute ago, that this neuron fires at the beginning, this neuron fires at the end of the track, and there is a sequence. Now you can ask whether this sequence in a novel task or a familiar task is similar during the waking state, when the animal is waking, resting at the end, or consuming a reward, or during sleep before or during sleep after. Doesn't matter how you make the template, there is a very strong connectivity. There is a very strong preservation of the sequences that are being generated under different conditions. And because it was already present prior the animal entering into a novel environment, we can call it a preconfigured dynamic. Now, this preconfigured dynamic can be very nicely demonstrated by looking at the firing rate distributions during a naturally occurring ripple or a synthetically generated ripple with light. And what we found is an astonishing strong correlations of both pyramidal cells and interneurons. That is, those neurons, they fire at this level during natural, spontaneously occurring ripples, they will have the same, pretty much the same probability to fire at the time when we artificially induce it. And this is really interesting because it happens in this region of the brain, the CA1 region of the hippocampus, where there are no recurrent excitatory collaterals, or very few, between pyramidal cells. And we have shown that this pattern is probably maintained by the intrinsic properties of the neurons themselves. Now we can go one step further and we can say, can we create such place cells synthetically? One way of doing that is we can stimulate with micro LEDs and activate a handful of neurons under one recording shank, but not under the other shank. So in this case, a bunch of neurons will be activated always in the same part of the maze. So then it's running back and forth, back and forth, back and forth, back and forth in a linear maze or a linear track. And then after some control trials, we make these artificial place cells, in this case, 1, 2, 3, 4, 5 trials. And then we will see what happens. And what will happen is that this is an artificial pattern. And there is during the stimulation, there is a very powerful excitatory gain. And then the neurons will maintain the place preference where they were stimulated. The interesting thing, of course, and the astonishing thing is that not every neuron does this. Only some neurons become place neurons, and we wondered why is this variability? And the answer is that only those neurons become place cells which already had the propensity, which already had a probability, a subthreshold or undetectable level of place feature at that particular part of the environment, at that particular segment of the track. Why? How can I say that? The reason why I can make this statement is because what we have done here is that we concatenated, we averaged all the neurons which with induced place fields. Then this would be a place field. This is the center of the place field. And this is after the induction. And we ask, can we see firing patterns that would be random anywhere in the track, or some lawful firing? And the answer is there is a lawful firing rate distribution. This is very weak. These neurons could never be classified as place fields because they fire sporadically 1 or 2 spikes per trial or even less. But when we add and average all the neurons, all their place fields related to the expressed place field, the artificially induced place field, then we can reveal that they already had a propensity, the probability to fire them. We can go even further and ask, if this neuron is firing in a particular part of the environment, we know that there are other neurons that are naturally fire in that particular environment or that particular segment of the track. Are the neurons that fire here in this particular aspect of the track, where the artificial neuron fires, can this relationship be deciphered prior to putting the animal or prior to trying to express a place field in this neuron? And the way how we can do that is that during sleep, we are looking for partnerships between the neuron in which we express the place fields and other neurons. And we find that this relationship is such that those neurons who were partners during sharp wave ripples during the preceding sleep, and they are the neurons that have overlapping place fields, similar place fields on the track. Now our new neuron that was already part of the assembly, but it was subthreshold, becomes suprathreshold and will become a place field in the area of the maze where the partnership dictated. So where is this pre-existence come from? One idea is that it's developmentally regulated. In order to address this question, a graduate student, Roman Hussar, did the following experiment. In embryonic ages, when neurons are developing, we can inject various, we can label neurons in various ways, and we can express channel rhodopsin, that is light sensitive opsins, in those neurons which are dividing at the moment when we use this method called electrophoresis. So these are examples when the neurons were labeled at embryonic day 13.5, 14.5, and so on. And you can see that their distribution is somewhat random, but somewhat lawful. And if you look at the depth distribution, we will find that the early born neurons are in the deeper layers. And as time goes by day, then the newborn neurons are more and more added to this layer from what we call the superficial layer. We can ask now with optogenetics, that is, we label neurons when they were born, and we wait until the animal becomes an adult, and then we are asking how these neurons that were born on a particular day behave and relate to each other relative to the other neurons. So this is a complicated method. It's measuring the spike transmission probability that I mentioned earlier. The important message here is that those neurons that were born on the same day will have a higher probability of innervating the interneurons compared to the different day born neurons. And this is just expressed here in a bar graph manner, showing that the same day neurons have a better chance of forming their own private subcircuits than neurons born on different days. The consequence of this is that when we just look at the cross correlation, the temporal cross correlation of neurons in a given session, for example, during sleep, we find that those neurons that are born on the same day, they like to fire together and more strongly with each other than with other neurons. And this is the same situation when we pick a sharp wave ripple and we ask, the blue ones are the ones that are born on the same day, and the red ones that are born on different days. And then we ask, what is the relationship between the same and different day neuron? And this is illustrated here. The same born neurons tend to fire together during sharp wave ripples significantly more than on neurons born on different days. It is true not only for sharp wave ripple, but also another pattern, a famous pattern of the hippocampus called hippocampal theta oscillations. And in this case, we ask whether the same born neurons are firing together on the same theta cycle. The answer is yes, they are firing at a higher probability. And this is true also for assemblies that are firing in the many neurons fire together, and the assembly membership is much stronger for those neurons that are born on the same day. So if this is an internal dynamic that it exists, how does it relate to something that we already know about the hippocampal neural features, such as the place fields? And the answer is that there is a strong relationship. So in this example, you can see that is a place neuron, or at least in this case is an assembly. There is another assembly that was born on the same day, another one that is born on the same day, and one happens to be fired on the place field of one assembly and the other one on the other assembly. And then this can be compared with a third assembly that has nothing to do with them, is that was born on a different day, or at least the members were born on a different days. And then we quantify it, and we find that indeed assembly membership is a predeterminant whether firing on the track will have overlapping place fields or not. This is just an example from the hippocampus, and we assume that this probably generalizes also to other features. In other words, the typical thing in the tabula rasa literature, I would say, is that neurons that are firing together, this is the Hebs rule, wire together. We may claim that, in fact, the opposite is true. Neurons that are born together will wire together, and therefore will fire together. All this leads to good news. We can reject the hypothesis that brain networks, at least in our case, hippocampal networks, are like slate. So where does this lead us? This leads us to another alternative. Namely, we can say that the reason, the way how patterns in the brain, sequences in the brain are generated is not necessarily done through imposition of the external environment on the hippocampal networks or brain networks. But the other alternative is that the brain is more like a spaghetti, that there are many, many, many patterns that are available. So the brain could be conceived, this preconfigured brain is like a Chinese dictionary for a European reader who doesn't understand any of the symbols. But with experience, certain symbols can be translated, can be linked to English sentences or English words. And by this grounding mechanism, by this experience, some of the available patterns, such as shown here with the thicker lines, become meaningful. And there are, of course, this is not a absolutely novel idea. Everything has history. And when you have these patterns, then some of these patterns can be concatenated, and most of the concatenation or grouping can occur with the help of inhibition or the coordinated action of inhibitory interneurons. To summarize what I would said, brain networks are preconfigured dictionaries filled initially with nonsense patterns, but with time, something happens, and learning is not a building up, de novo, from scratch process, but in fact, it's a matching process between a pre-existing pattern and experience. And so meaning is acquired by adding a new utility, a new significance to pre-existing patterns that is something that adds something useful to their animal. And of course, the most important conclusion is that the psychophysical Weber-Fechner law is a consequence of this preconfigured log dynamic organization. I know that what I told you is a little bit complicated. If you are interested in more details, I recommend a more extensive treatment of the same topic. And that's my recommendation. Thank you for your attention.